type of macromolecule is nucleotides. Nucleotides form a very important large molecule called DNA. DNA stands for deoxyribonucleic acid. DNA holds the chemical message for producing every part of the organism. It regulates life by carrying the directions for growth, repair, and replacement of cells and their parts. DNA is the genetic material that carries all the instructions to make an individual. DNA is packaged tightly in the cell in the form of chromatin. When it is unwound and stretched out, it looks like a ladder that has been twisted. This is called the DNA double helix. It is double because it is made of two strands of DNA that connect and look like a ladder. It has a helix because it has a natural twist to it that causes the molecule to spiral. DNA is a macromolecule. That means it is a large molecule that is actually a polymer. The monomer of DNA is a nucleotide. A nucleotide is made of 3 parts: 5-carbon pentose sugar deoxyribose, 1 to 3 phosphate groups, PO43-, nitrogenous base, adenine A, thymine T, cytosine C, guanine G. A sugar pentose plus a nitrogen base is a nucleoside. A sugar pentose plus a nitrogen base plus 1 to 3 phosphates is a nucleotide. There are 4 nitrogen bases that exist in DNA: guanine G, adenine A, cytosine C, thymine T. The nitrogen bases are found facing the middle of the double helix and form the rungs of the DNA ladder. The sugar, a 5-carbon pentose, and phosphate groups make up the sides of the ladder. The sides of the ladder are referred to as the sugar-phosphate backbone or the phosphodiesterase backbone of the DNA double helix. Each nucleotide is attached to the next nucleotide through the sugar-phosphate backbone by a hydrolysis reaction forming a phosphodiesterase linkage. When the oxygen in the hydroxyl group is missing, the ribose sugar is called deoxyribose. In RNA, there is an oxygen atom, so the sugar in RNA is called ribose rather than deoxyribose. It also has uracil U instead of thymine T. To recap from Unit 1, the sugar-phosphate backbone makes the sides of the ladder, and the rungs are made of the 4 nitrogen bases. Their names are often represented by the letter of the base name, A, C, G, and T. The actual ladder itself is made of 2 strands of DNA, which is why it is called a double helix. Each strand attaches by the nitrogen bases that pair in the middle, the rungs of the ladder. A pairs with T, held by 2 H bonds. G pairs with C, held by 3 H bonds. On each strand of DNA, there is a 3' end and a 5' end. Genes are units of heredity that are passed on from parent to offspring. When a cell is preparing to divide, the genetic material containing the genes becomes visible as chromosomes. A gene is made of introns and exons. Exons are sections of the chromosome that code for proteins. Introns are non-coding sections of the chromosome. DNA is folded and coiled around proteins called histones. Genes for specific traits may exist in alternative forms called alleles. Alleles are slightly different forms of the same gene. Mitosis allows the nuclear material in the cell to be divided into daughter cells. Just prior to mitosis, the nuclear material in the cell is duplicated. This process is called DNA replication. DNA replication allows both sides of the DNA to be copied simultaneously. Once the 2 strands are separated, this is referred to as a replication fork. Let's look at the enzyme proteins involved in the process. Gyrase. This is a large protein that unwinds the DNA and releases stress on the molecule. Helicase. This is a large quaternary protein, about 6 tertiary structures that can unzip DNA by separating H bonds. Single-stranded binding proteins, SSBs. This is a quaternary protein that prevents the 2 separated strands from reannealing. DNA polymerase 1 or 3. These are a group of quaternary proteins that read single-stranded DNA in the direction of 5' to 3' and assembles nucleotides in the new strand from 3' to 5'. It can read and extend a nucleotide sequence, but cannot start one. There are DNA polymerase 1, DNA polymerase 3, DNA polymerase A, etc. Primase, an RNA polymerase that makes short RNA primers to assist DNA polymerase so it has a place to start the replication of nucleotides. DNA polymerase 1, RNase H, a protein that changes the RNA primer to DNA by hydrolyzing at the 2' carbon. DNA ligase joins short sections of DNA together, forming a continuous strand. The process. Gyrase unwinds the double helix, helicase unzips the DNA, and SSBs prevent the 2 strands from rejoining. A RNA primer is formed by primase so that DNA polymerase 3 has a starting point. DNA polymerase 3 assembles nucleotides 3' to 5' but reads the template 5' to 3'. Due to this, one strand can be formed continuously, and this is called the leading strand, and the other strand must be formed in short segments as helicase unwinds the DNA. This is called the logging strand and is made in short segments called Okazaki fragments. DNA polymerase I will convert the RNA primer into DNA. DNA ligase will join the fragments of DNA by connecting nucleotides through phosphodiester linkages. Replication terminates when DNA polymerase III reads telomerase. These are repeating sequences of DNA that get removed and become shorter after each time DNA is replicated. Finally, there is a proofreading of the replicated strand to catch any errors. This is done by another DNA polymerase. Each strand will not look like the template it is copying from, but rather the antiparallel strand of the double helix. Transcription and translation. Transcription, DNA to RNA. Translation, RNA to protein. The DNA is like a reference library with books and material. You are allowed to borrow, sign out, but you are not allowed to borrow, sign out, but you can make a copy of several pages. The copy that is made is called messenger RNA or mRNA. The process of creating the message is called transcription. This process of translating the message into a functional protein is called translation. Messenger RNA is single-stranded RNA that is a copy of a portion of the original DNA strand. It carries the RNA copy of the DNA code for one or more genes. The mRNA carries a genetic message in a series of codons. One codon codes for one amino acid. Ribosomal RNA is single-stranded RNA that in most species is made of two subunits. The subunits work together to make a functional ribosome that is capable of reading mRNA from 5' to 3' and assembling amino acids to make a functional protein. Transfer RNA is single-stranded RNA that is capable of attaching to an amino acid at the top near the acceptor stem. Transcription is where a copy of the DNA is made. The process of transcription can be divided into 3 steps that make it easier to understand: initiation, elongation, and termination. The main enzyme in the process is RNA polymerase. In the initiation step, RNA polymerase binds to the promoter region that is upstream to the gene. Once the RNA polymerase clears the promoter region, it starts assembling single-stranded mRNA, 5' to 3' in elongation. It unwinds, unzips, and copies one strand of the DNA, which is referred to as the template strand. T will be replaced by U. Termination occurs when RNA polymerase recognizes the termination sequence and releases the new mRNA. Modification of the message. The mRNA may need to be spliced, much like a movie film, to delete unwanted excerpts. The enzyme that does this is large and called a spliceosome. A spliceosome removes non-coding sections of the message called introns and keeps the coding selections of the message called exons. Translation. 3 bases code for an amino acid. 3 bases are called a codon. This third amino acid is called the wobble position. This allows for more than one codon to code for an amino acid in the case of error. The expression of genes must be controlled by the cell. Transcriptional control is the cell can control which genes are transcribed and can slow down or speed up the production of mRNA. Post-transcriptional control, the cell can make mRNA and break it down before it is translated, or shorten or lengthen the poly A tail to regulate how quickly the message is broken down. Translational control is the cell controls how quickly the message will be translated and how long before enzymes in the cytoplasm break down the mRNA. Post-translational control, the cell may regulate the modification of a protein, controlling the rate at which it becomes functional, and also control the time it can remain functional. An operon is a group of genes that include structural genes as well as operator and promoter regions on the DNA. Operons are regulatory units in prokaryotic cells. The IAC operon is an example of transcriptional control operon in E. coli bacteria. This enzyme is located on a cluster of genes called an operon. When there is not enough glucose in the cell, cyclic AMP, cAMP, will accumulate in the cell. The TRP operon is another example of transcriptional control in prokaryotes. Gene regulation in eukaryotes offer more opportunities and levels of control than in prokaryotes. A point mutation is an error at a single point in the DNA. Some point mutations do not affect the protein formed at all. These are called silent mutations. It may be due to deletion of a base pair or the insertion of a base pair that can cause a frame shift mutation. That causes a shift so the codons are now read differently and the entire sequence is thrown off. Another type of substitution mutation causes protein synthesis to stop. Nonsense mutations result when a substitution of base pair forms a stop codon. Other mutations may be missense mutations. This type of mutation can be harmful because the protein can be altered. Other mutation types. Deletion. A part of a chromosome is missing. Duplication. A part of a chromosome is duplicated more than once. Inversion. A part of a chromosome is inverted on the same chromosome so the genes on that segment are in a different location. Insertion. A part of a chromosome is inserted into another chromosome. Translocation. A part of a chromosome is translocated to the end of another chromosome. Non-disjunction is an error that occurs in meiosis. Down syndrome is an example of non-disjunction of chromosome 21. Mutations can be caused by chemical mutagens and physical mutations. Many chemical mutagens are carcinogens, meaning they are cancer-causing. For example, high-energy radiation such as UV light and X-rays can cause H-bonding nucleotides of the same strand, forming dimers. DNA must be repaired regardless of mutagens. Reproductive technology is the use of technology to assist or enhance reproduction in humans or other organisms. Cloning is the creation of an organism that is a genetic copy of another. Dolly was created through a process called somatic cell transfer. The nucleus of a cell from an adult female sheep was extracted. Biotechnology utilizes biological processes such as fruit ripening or living things which are genetically engineered to create specific products for industries such as agriculture and medicine. Stem cells are undifferentiated cells. Undifferentiated cells are cells that have not yet specialized and turned into a specific type of cell. Recombinant DNA is artificial DNA made from combining the DNA of two different species. Plasmids are segments of circular DNA found in bacteria that are naturally occurring and separate from the bacteria's DNA. Recombining DNA involves using bacterial plasmids and special enzymes called restriction endonucleases. Restriction endonucleases are a class of enzymes that are found in bacteria and viruses that can cut double-stranded DNA. The enzyme cuts the DNA and creates a sticky end where a gene can be inserted. Scientists use these enzymes to cut the DNA of the plasmid-inserted gene, often of a different species of their choice, into the plasmid and then reseal the plasmid. This recombined DNA is recombinant DNA, rDNA. There are enzymes, restriction endonucleases. There are enzymes that are found in prokaryotic cells that are used by these organisms to cut out foreign DNA. This class of enzymes recognizes specific sequences on the DNA and cuts the DNA at that sequence known as the restriction site. The fragments that are cut are sticky. That means they can form base pairs with other pieces of DNA that have the complement. Creating many copies of a specific gene or portion of DNA is called DNA amplification. This can be done by several methods: bacterial vectors and polymerase chain reaction. Bacterial vectors. The desired gene can be inserted into a plasmid using restriction enzymes, and then the plasmid, now recombinant, can be inserted into a bacterial cell. The bacterial cell then multiplies and makes many copies of the gene. This type of amplification is used for large sequences of DNA. Polymerase chain reaction, PCR. In this process, the desired gene is replicated using DNA polymerase. First primers are used to complement the DNA sequence, so DNA polymerase has a place to start. The DNA is heated to separate the double helix and then cooled. DNA polymerase now starts adding nucleotides and replicates the gene. Gel electrophoresis is used to separate molecules by mass and charge. DNA analysis is used in criminal investigations and has a high degree of accuracy. DNA sequencing is done to determine the nucleotide sequence of portions of DNA. The process for doing this is known as chain termination sequencing. Societal concerns with biotechnology according to ThoughtCo. Biotechnology is the use of living systems and organisms to develop or make products, or any technological application that uses biological systems, living organisms, or derivatives to make or modify products or processes for specific use. New tools and products developed by biotechnologists are useful in research, agriculture, industry, and the clinic. There are 4 main societal concerns in the biotechnology field. Here's a closer look at these concerns in this ever-changing field, along with some of the main reasons why we use this controversial science. 4 societal concerns with biotechnology. There are 4 main concerns we, as a society, have when it comes to this ever-advancing field. Harm to the environment. This concern is perhaps the most widely cited by those opposed to GMOs. It is very difficult to predict what will happen in an ecosystem where a new organism has been introduced, whether genetically modified or not. Take weeds, for example. If farmers introduce a herbicide-resistant marker into a plant, there is the possibility those traits may be transferred to a weed, making it resistant to herbicides as well. Bioterrorism. Governments are worried terrorists will use biotechnology to create new superbugs, infectious viruses, or toxins for which we have no cures. According to the CDC, bioterrorism happens when viruses, bacteria, or other germs are released intentionally to inflict harm on or kill people, plants, or livestock. The agency says the most likely agent to be used in an attack is anthrax. a serious disease caused by a bacteria found naturally in soil. The use of viruses and diseases as a weapon in warfare has been well documented in history. Native Americans were infected by the British Army in the 1760s when they were given blankets from a smallpox hospital. During World War II, Japan released bombs on China containing fleas infested with disease. In modern times, bioterrorists are able to transfer diseases and viruses through explosives, food and water, and even aerosol sprays. But the use of biotechnology as a weapon was banned by the Geneva Convention. Laboratory production safety. It's hard to protect yourself if you don't know what you're working against. Some new technologies, usually non-biological such as nanoparticles, make commercial production lines before they have been sufficiently tested for safety. There is also concern about technician safety in laboratories, even under secure conditions, when working with organisms of unknown virulence. Ethical issues. Besides the age-old debate over whether cloning genes is sacrilegious, innumerable ethical questions arise over the appropriateness of licensing genetic inventions and other IP issues. In addition, the construction of genes from scratch, the first artificial gene was actually synthesized in 1970, means we might someday be able to create life from a chemical soup, which will most certainly go against the ethical or religious beliefs of a significant number of people. There are also other ethical concerns, including when scientists use humans as clinical trial subjects. People will often try anything in order to help combat illness or disease, especially when there is no known cure. How do scientists protect their subjects when they are unsure of the results or side effects of any study? Activists are critical of the use of animals as test subjects in biotechnology. Scientists may manipulate animal genes, all for the benefit of human lives. The animal, therefore, becomes nothing more than a piece of property rather than a living being. Why is it used? We use biotechnology to make medicines and vaccines to fight diseases, and we are now turning to biotechnology to find alternatives to fossil-based fuels for a cleaner, healthier planet. Modern biotechnology provides breakthrough products and technologies to combat debilitating and rare diseases, reducing our environmental footprint, feed the hungry, use less and cleaner energy, and have safer, cleaner, and more efficient industrial manufacturing processes. More than 13.3 million farmers around the world use agriculture biotechnology to increase yields, prevent damage from insects and pests, and reduce farming's impact on the environment. Growing biotech crops can also help lower the cost of production, cutting back on expenses like fuel, water, and herbicides. This is especially important for farmers who can't afford the high cost of agriculture and can help farmers in developing nations. A changing field. The field of biotechnology is fast-paced and rapidly changing. Often the pace at which new technologies are developed far exceeds that of regulatory change and adaptation, which generates significant bioethics issues, especially since many of the new developments are those that impact human lives directly through what we eat, drink, and the medications we take. Many scientists and regulators are very aware of this disconnect. Thus, the rules for issues such as stem cell research, patenting genetic inventions, and new drug development are constantly changing. The relatively recent emergence of genomics and methods for creating artificial genes present new threats to the environment and the human race as a whole. The bottom line, biotechnology is a constantly evolving field of science. Although it has many benefits, including lowering our environmental footprint and helping treat disease and illness, it doesn't come without its disadvantages. The four main concerns revolve around ethical, safety, bioterrorism, and environmental issues.